All right, well, welcome back. We are going to close out this session with Dr. Georgia Purdom. She is a geneticist. She graduated with a PhD in molecular genetics from The Ohio State University. Yeah, I got some excitement from that one. Yeah, her graduate work focused on genetic regulation of factors important for bone remodeling. That's exciting, isn't it? Yeah, that's right. Well, following graduation, Dr. Purdom served as a professor of biology for 6 years at Mount Vernon Nazarene University in Ohio. She has been on staff for a number of years. She is an accomplished speaker, writer, and researcher here at this ministry, and she is the chair of the editorial review board for content here at the ministry. And I would like to welcome Dr. Georgia Purdom.
Well, thank you. It's a pleasure to be here. I guess I should say go Bucks, okay, since everyone seemed excited about Ohio State University. All right. Well, tonight we're going to be talking about the wonder of DNA. And, you know, there is so much information on this topic that it was really difficult for me to put this in 1 hour presentation. In fact, you will see in your outline that there are some things that we will not discuss tonight because as I reviewed this, I realized it's too long and I need to get it down into an hour. And really what we're going to be doing is looking at this from 30,000 feet without going into extensive detail on any one subject, but hopefully gaining a great appreciation for the complex design that is there. And we're mainly going to talk about human DNA because that is where most of the research has been done, and I figure that's probably of a lot of interest to us since we are human. And what I'm going to do first of all is start with sort of a short primer on DNA so that you'll understand some of the terminology that I'm going to be using throughout the presentation. And so let's talk about some basic facts about the human genome. There are approximately 3 billion base pairs that make up the human DNA in the nucleus, and it's organized into 23 pairs of chromosomes. It has approximately 20,000 genes. Now, genes are the instructions for how to make the proteins, and it's whoops, one of my points is missing there. Let me see if I can get that back. I'm not sure where it went. Well, what I was going to say was it's a twisted double helix, okay? It forms kind of a ladder, and it's composed of 4 bases: adenine, cytosine, thymine, and guanine, A, C, T, and G. And you can see that here on the slide. We have the twisted double helix, and we have adenine always binds to thymine, cytosine always bonds to guanine, and you probably are very familiar with that. You've seen pictures of that before. And so next what I'm going to do is show you a short video that really gives us an idea of the size of the genome and how much of it actually codes for proteins.
Think of how many letters fit on a single page. And now imagine a stack of pages 300 feet tall. That's how much information is stored in the DNA inside every human cell, the entire human genome. If you sort through the 3 billion letters that make up the human genome, you find some surprising things. Only about 1% of the 3 billion letters directly codes for proteins. Of the rest, about 25% make up genes and their regulatory elements. The function of the remaining letters is still unclear. Some of it may be redundant information left over from our evolutionary past.
Okay, now, first of all, okay, you know some things I'm going to disagree with, right? But I would disagree, though, that when she says this is how much information is stored in our DNA, the stack really reflects just the linear sequence of DNA. If you wrote out all 3 billion bases on those papers, that's what you would end up with, okay? That stack of papers 300 feet high. But DNA is a 3-dimensional structure, and so there literally is what we're going to talk about tonight, layer upon layer upon layer of information. It's not just about the linear sequence of the DNA. Now, she goes on to say that a lot of DNA, we just simply don't know what it does. And she didn't say junk DNA, but that's what it's affectionately referred to as. And she says it's possibly a leftover from our evolutionary ancestors. And we're going to talk about why that is really not the case. So the central dogma of molecular biology is that DNA, of course, has the information on how to make the proteins, and it is transcribed into what we call RNA. RNA. It's very similar chemically to DNA, but it's kind of like a Xerox of the DNA. DNA stays in the nucleus, as far as we know. It doesn't leave. And so you kind of have to make a Xerox of it that can leave the nucleus and then be translated into a protein, okay? And that is what makes up, what does mainly the structure and functions in our body. They're the workhorses, basically, of the human body. And in these processes alone of transcription and translation, there is a lot of complexity in design that we could go into more detail about. I'm not going to. If you want to know more about that, I encourage you to get my DVD, Wonder of the Cell, where I talk a little bit more about the detail there. But I do want to show you some short clips that just showcase these processes so you can, again, kind of get an idea of what's going on here in the cell. And the first one is going to show us transcription.
What you are about to see is DNA's most extraordinary secret: how a simple code is turned into flesh and blood. It begins with a bundle of factors assembling at the start of a gene. A gene is simply a length of DNA instructions stretching away to the left. The assembled factors trigger the first phase of the process, reading off the information that will be needed to make the protein. Everything is ready to roll. 3, 2, 1, go. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the 2 strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA.
Okay, like I said, that's a complex process in and of itself. And they're not even showing you everything that's going on, by the way. This is the simplified version of it, and it's much more complex. Now, what happens after we get that RNA? We have to translate it. The cell has to translate it into a protein. So here's how that process works.
When the RNA copy is complete, it snakes out into the outer part of the cell. Then all the components of a molecular factory called a ribosome lock together around the RNA. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. Inside the ribosome, the RNA is pulled through like a tape. There are different transfer molecules for each of the 20 amino acids shown as small red tips. The code for each amino acid is read off the RNA 3 letters at a time and matched to 3 corresponding letters on the transfer molecules. The amino acid is added to the growing protein chain, and after a few seconds the protein starts to emerge from the ribosome.
Okay, now that's what happens for about 2% of our DNA. Right? It's codes for proteins. At least it goes to the protein level. But what about the other 98%, right? That seems like a lot of DNA. And a lot of that is involved in the regulation of these processes. So where we have things like we have to have transcriptional control, and I thought that was going to show up on the screen, but it's not for some reason. So we have transcriptional regulation, where it's transcribing from DNA to RNA. We have to regulate that. We have to regulate going from, once we get the RNA transcribed, there's all these things that have to happen to it, so that's post-transcriptional regulation. Then to go from RNA to protein is translational regulation, and then there's post-translational regulation, and there's all these levels that have to be controlled. And so where is that information for that regulation? Well, some of it is in the so-called junk DNA, which we're going to be talking about. Some of it is actually in the genes themselves, and some of it is actually in the 3D, the three-dimensional structure of the DNA. And we're going to talk about all 3 of those areas today. And it's really, we're going to be talking about the design and the regulation of DNA. And the reason I chose that, because you have to choose one part, okay? Because there's so much design here, because I think it beautifully illustrates the design in DNA. And you know, as we look at the complexity of DNA, which falls into the category of observational science, which is here and now science. Okay, so what I'm going to be showing you tonight is a lot of observational science. We can see it, we can observe it, we can know those things. And we need to keep in mind, though, that who or what we attribute the complexity that we observe in DNA to really depends on our starting points. Now, that falls into the category of historical science, right? That's something that happened in the past. The origin and complexity that we observe in DNA is an event that has happened in the past and is very much dependent on our authority for knowing about the past. Because the evolutionists believe that the complexity we observe is not designed, but rather what they call apparent design. It looks designed, but it really isn't. And that, again, why do they say that? That's based on their starting points, their authority for knowing about the past, and they believe that man's ideas about the past, that all life has come about by random chance over millions of years. In this next video, I'm just going to show you a short clip of this, illustrates this idea well in talking about Watson and Crick's discovery of the structure of DNA.
It dawned on Watson and Crick that this complementary copy of the code was the key to understanding how genetic information could be passed on. So the essence of life could be explained with chemistry. There were no mysterious life forces.
Okay, now notice what she says there. She's talking about observational science. We can observe how DNA base pairs, we can observe its structure, and then she goes into talking about historical science, right? Well, there's no mysterious life forces. This all just came about by chance. And even though they say this, though, they realize that what they are saying seems very contrary. Leslie Orgel, who's a British chemist, stated this: "It is genuinely surprising that an organism that has evolved by random mutation and natural selection appears to be designed. The idea is contrary to intuition. It is true, but to many it will always remain ridiculous." And I agree with that, because living things, DNA, obviously appear designed, and thinking otherwise, he said, I don't agree with this part, it's contrary to intuition, right? So, because that's not our normal everyday experience. If something looks designed, it is. So why do evolutionists then rule out that possibility from the start? Why is it not really designed? And this next quote comes from Fred Hoyle and Chandra Wickramasinghe, who are British astronomers, and they stated this: "To involve purpose is in the eyes of biologists the ultimate scientific sin." And it is. The revulsion which biologists feel to the thought that purpose might have a place in the structure of biology is therefore revulsion to the concept that biology might have a connection to an intelligence higher than our own. Because design means purpose, and purpose means an intelligence, right? It means something else. And not random chance, but rather a designer. So as we study DNA today, the observational science, we'll see that the design there confirms and is consistent with the historical science based on God's word and the result of God's intelligence, and is inconsistent and does not confirm that DNA is the result of random chance evolutionary changes over eons of time. Now, doing the presentation, or doing the research for this presentation and learning more about the amazing design in DNA made me realize even more that this is not an issue of the evidence, because the evidence is clear. It kind of blows my mind sometimes as a geneticist how other geneticists who don't know the Lord, who don't start with God's word, can look at this exact same thing and say something so different. But what it comes down to is this is an authority issue, and ultimately a heart issue in which many scientists want to be their own God, right, instead of believing in the one true God who designed and created our DNA. Now, there's three major types of design that we're going to discuss tonight, and I categorize them according to whether they're sequence design, structural design, or what I call adaptive design. Now, again, something to keep in mind for each of these categories of design. I'm only discussing a very, very small fraction, okay, of the design that is actually present in the DNA. So the first one I'm going to talk about is sequence design, and we're going to first talk about hidden codes. Now, by hidden codes, I don't mean Bible verses hidden in DNA, okay? Not there. All right, now, something else, okay, so don't get that impression. I don't want to get the wrong impression. So the first hidden code we're going to talk about is something called duons. And duons, this research is just, like, literally came out last year. And duons are codons that are found in genes. Now, let me explain what a codon is. You actually saw it, you just didn't realize you were seeing it in those videos. A codon is in the DNA, and it codes for, it's called a codon because it codes for an amino acid in the protein. And it's 3 bases, okay? 3 bases in the DNA, gets transcribed in RNA, and then gets read by the ribosome, which is a little protein factory, reads it, inserts amino acids for that particular codon. Now, they found out that some codons have dual purposes. They were thought for many years just to specify which amino acid was used in the protein. But now they found out that some of them actually specify where factors bind to and regulate the expression of the gene. Now, this is very anathema, okay, to molecular biology, because originally it was thought the protein coding region and the regulatory region were completely separate, okay? Physically and functionally, they were 2 completely different things. Whereas now we're finding out, no, they can actually be doing 2 things. And it applies to about 15% of human codons, which is literally millions and millions of bases. And really with these findings, it becomes even more difficult to say any mutation is neutral. Because scientists used to think that as long as a mutation in a codon doesn't change the amino acid it codes for, because multiple codons can code for the same amino acid, then the mutation was neutral, right? It didn't affect the amino acid, so it's neutral. But if codons have dual purposes, then the mutation could be affecting its ability to bind to a certain factor and regulate the expression of the gene, even if it doesn't ultimately change the sequence of the protein. So while a mutation might be beneficial, or neutral in relation to one function, it could be detrimental to the other function. You see what I mean? Because they got dual functions. So you start messing with it, chances are it's going to be detrimental in some way. And there's obviously no net gain here needed for evolution to occur. So it becomes even more implausible to believe that random mutations over time have generated the complexity and diversity of the organisms that we see today. This is clearly design and not disorder. And one of the researchers involved in this finding said this: for over 40 years, we have assumed that DNA changes affecting the genetic code solely impact how proteins are made. Now we know that this basic assumption about reading the human genome is half of the picture. I think half of the picture is a gross understatement, okay? It's probably 99.9% of the picture. I mean, that's how much of it we're missing. And you know, why did they assume that the human genome was so simplistic to begin with? Again, because they are starting with evolutionary assumptions. It would predict relative simplicity, right? Since everything came from chance by a simple common ancestor. So the level of complexity is really inconsistent with what they would predict, but totally consistent with what we would predict, starting with God's word and God as the designer and creator of our DNA. Now, another hidden code I want to talk about is something called dual coding genes. And dual coding genes is when one gene can encode 2 different proteins. Now, again, this is like molecular biology heresy, right, if there is such a thing. Because I learned in graduate school and in college that one gene equals 1 protein, right? That's what you're always taught. That's how it goes. But now we're finding that's not the case, the more that we explore this. And when I say 2 different proteins, I mean 2 different structurally unrelated proteins. Now, they're not different versions of the same protein. That's done by another process called alternative splicing, which we're not going to get into. But that's another mechanism to get different versions of the same protein. No, these are 2 completely different proteins. And right now they have about 40 candidates for what they believe are dual coding genes, and there may be more. Again, they've just started studying this. So this just shows you kind of an example here. So on the top there, you have a gene, the big long one there, that produces a protein that's called a G-alpha subunit, okay, a G protein alpha subunit. It's involved in cell signaling. But they found out that this same gene, a smaller portion of it, encodes a protein called ALEX. Now, we don't know exactly what that protein does, but we know it exists, and they're still trying to figure that out. But they're very structurally unrelated. They're not doing the same things. They're 2 totally different proteins. And the researchers on this really think they've underestimated how many of these there are because they used extremely conservative parameters when trying to find these. And so it may help explain why we have 20,000 genes, but 200,000 to 300,000 different proteins. Something doesn't match up. So there's got to be more than 1-to-1 here. Now, dual coding genes are extremely problematic for evolution. Why? And here's what the authors of this paper said. Dual coding is a costly arrangement because it limits the flexibility of amino acid composition. A silent change in one frame that codes for one protein is almost always guaranteed to be amino acid changing in the other. Now, so in other words, based on evolutionary assumptions, this doesn't make sense because it constrains DNA. You can't have changes because if you change one thing and it might be beneficial, you're going to change another thing that's going to be detrimental. And that doesn't get you to where you need to be, so to speak, for evolution. So less is available for change because, like I say, you might be beneficial here, like the protein, but then you affect the regulation or something like that, or you affect the other protein, which is detrimental. Now they say, although counterintuitive, once again, yep, it's counterintuitive, right? Because it's totally inconsistent with the evolutionary assumptions, which would predict that, again, you shouldn't have these things, but totally consistent with what you would predict with God's word and God as the designer and creator of our DNA. Now, the next type of sequence design that we want to address is something you all probably are familiar with, and that is the term junk DNA. So approximately 98% of human DNA does not code for proteins, okay, as far as we know. Could be saying something different in 10 years, but as far as we know, right now, it doesn't code for proteins. And they are considered to be, as you heard in the video, genetic fossils or leftovers from our evolutionary past. So just like you can find fossils in the ground, you have genetic fossils, right, in your DNA. And there are many different types, and these are just three of the major categories. Non-coding RNAs means they're made into RNA, but they're not made into a protein. Repetitive sequences, and what are called pseudogenes. And we'll talk about those in a little bit more detail later. But this little chart here shows you, if you look at the little green wedge, okay, that says protein coding gene 2%, right, that's all that codes for proteins. The rest of that is junk DNA. Different categories of junk DNA. That is the composition of the human genome. Now, Susumu Ono, who was a geneticist back in the 1970s, actually coined the term junk DNA, and he stated this: "The earth is strewn with fossil remains of extinct species. Is it a wonder, too, that our genome is filled with the remains of extinct genes?" Right? They're fossils. The thinking was that as we evolved up the evolutionary tree, that genetic information that was no longer necessary or useful, say information on how to make fins or a tail, began to degrade and accumulate mutations, but it was still part of our DNA, which from an evolutionary standpoint is kind of odd as well, because if it wasn't useful, it wouldn't be selected for, so why is it still there? But nonetheless, they would just say, not enough time, right? That's always the key for evolutionists. But since the release of the sequence of the human genome in 2003 and discovering that we only had around 20,000 genes, there's been a lot of interest in studying the junk DNA. And a lot of that research has been done in particular by the ENCODE project, which stands for Encyclopedia of DNA Elements. It involves many scientists and research facilities around the world, and they have published now dozens of papers in scientific journals. And I've actually written about this several times on our website, so if you want to find out more about ENCODE, just type in ENCODE and Purdom, and you can find that. So the first phase was completed in 2007, and they studied 1% of the human genome, the junk. Nearly all of it, they found, was transcribed into RNA. So even though it wasn't going to go on and become a protein, it was made into RNA. So maybe it has some function as RNA, which again, no one ever thought about, that there were functional RNAs, okay? The RNA did things like this. And Tim Hubbard, who was with the Wellcome Trust, who helped work on the ENCODE project, said, "We are now seeing the majority of the rest of the genome is active to some extent. We may not know what it's doing yet," they said, "but it's doing something." Francis Collins, right, the director at that time of the National Human Genome Research Institute, said there's a lot more going on than we thought. And Eric Green said the take-home message is, oh my gosh, this is really complicated. I love it. Yeah, it's complicated, you know, which is screaming design, right? Screaming a designer. So phase 2 was completed in 2012. And they studied the remaining 99% of the human genome. Now, this is no easy task. It might seem like that, but you're talking 3 billion bases. Okay, that's a lot. They found that 80% of it is functional because it has what they termed as specific biochemical activity. It's doing something. And again, it keeps advancing. So they found out it was transcribed into RNA. Now they're showing a little bit more about what its activity is, and we're going to talk about that in a minute. And Tom Gingras, who is an ENCODE researcher, said almost every nucleotide is associated with a function of some sort or another. And we now know where they are, what binds to them, what their associations are, and more. Okay, so they found out a lot more details. They're starting to fill in the blanks. And Ewan Burney, who is the lead analysis coordinator on ENCODE, said it's likely that 80% will go to 100%. 100% functional. 100% doing something. It's obvious that we need a new name for junk DNA. I am so tired of saying junk DNA, because it's not. So again, we're seeing clearly design and not disorder. Now, phase 2, like I said, was completed in 2012, and what they found was that these functions are mainly regulatory. Big surprise, right? I mean, we are extremely complex beings. We are going to have a lot of regulation for these things. You've got the same DNA virtually in each cell of your body. Okay, but yet your liver cells have to do something different than your skin cells versus your brain cells. Think about the complexity that's required in the design to make that happen. And phase 3 has just begun. So when I give this talk in a few years, we'll talk about phase 3. And they've begun to study exactly what that function is in more detail. Now, what they found, too, with the 2012 study is that some variations in junk DNA have now been linked to disease. And so that study has really opened the door for understanding more how mutation may be involved in disease. So for example, they've just this year found out that it's non-coding DNA, the junk DNA, has been implicated in type 2 diabetes. It's been linked to cancer, schizophrenia, Hodgkin's lymphoma, heart disease. Okay? Mutations in places that they didn't think would have any effect, right? Because it's neutral. DNA, it's junk. It's not important. Well, it turns out it is important, and this is a big indicator of that. Now, there's several major categories of junk DNA, as I mentioned earlier, and one of those is what we call non-coding RNAs, and they function as RNA. They don't become a protein, but they have a function as RNA, and we'll talk about that in a minute. Then there's repetitive sequences. Do you realize that 50% of human DNA just says the same thing over and over again? Almost every single chromosome in the human body, almost 50% of it is repetitive DNA. Same sequences over and over, and we're going to talk about why that's important. And then pseudogenes. Now, these are non-functional copies of functional genes. They look a lot like a functional gene that we have, but they've got some mutations, so they maybe don't make a protein, or the protein's different in some way. And again, those were always thought to be non-functional. Okay, now, what are then some of the functions of junk DNA? And the biggest one, again, by far, is regulatory functions. And one of those regulatory functions is what we call gene silencing. And what it is you form this is true for the non-coding RNAs as well as the pseudogene. That RNA that's produced will pair with RNA that codes for proteins and stop it from being able to be translated into a protein. Now, I want to show you a picture of this, and let me tell you, I had to really scour the internet for a simple enough picture to show. It was so hard. I was about to lose hope. But I found this one because I just wanted to illustrate this really simply. So the top here where it says messenger RNA, okay, that's the RNA that gets translated into a protein. But what can happen is you can have these non-coding RNAs, which is called microRNA there on the screen. That's a form of non-coding RNA. It doesn't become a protein. But what it does is it's close enough in sequence or can bind to a part of the messenger RNA, and when it does, it can't be translated into a protein anymore. The messenger can't relay its message, shall we say. It can't be translated into protein, so it silences the gene. And there are so many of these. And again, it's part of that regulatory process. Now, another thing they do is they fine-tune transcription and translation to make it just right. Okay, so you get the right stuff at the right time under the right conditions. They format DNA. See all those repetitive sequences that people thought were junk? Well, it turns out they may have a lot to do with the structure of DNA. Okay, how DNA, and I'll show you this a little bit later. DNA, you got 6 feet of DNA, okay, in each cell. You got to package it up. really small, okay, to get it to fit inside the nucleus. And so these repetitive sequences probably have a very large role in that. There's been studies that indicate that they're involved in winding it up. So they're a structural component in that sense, in helping the DNA become small enough to fit inside the nucleus. And pseudogenes themselves may be involved in generating genetic diversity. I read about this when it comes to antibody formation, because we have to make tons and tons and tons of different kinds of antibodies throughout our lifetime. How do you get all that diversity from the few genes that we have for that? Well, part of it may be through pseudogenes. So junk DNA is not junk. It has a function. It is important. And it seriously constrains evolution, just like the hidden codes, because you can't do anything and everything, because for sure you're going to mess up something, okay? And that's the problem. And again, many have thought of junk DNA as a warehouse, right? That mutations can occur leading to the formation of new genes. But how plausible is that if most of it is functional and is doing something? You can't just pull stuff out and make something new, okay? Because you're going to damage something else. It's clearly design and not disorder. So then, have evolutionists given up on the idea that junk DNA is not junk? And I think some have, as we've seen evidence by these papers that I just showed you, but many evolutionists are still not willing to concede that junk is not junk. And John Timmer, who has a PhD and works in the molecular and cellular biology department at UC Berkeley, said this: "Personally, I fall into the it's all junk end of the spectrum. If almost all of these sequences are not conserved by evolution, and we haven't found a function for any of them yet, it's hard to see how the none of it's junk view can be maintained." Now, this was after ENCODE released its results. So he could see what they found, and he said, nope, it's still junk. Now, the argument he's making that when they did the study in 2007, and they compared lots of different mammals' junk DNA, okay? So dogs, humans, mice, cats, all of that. 23 different mammals. And what they found was that about 50% of the junk DNA was different, okay? It wasn't the same. Now, that's not how evolution works, right? If something's important and functional, it should be conserved as you go up the evolutionary tree. And so, because that's how evolution works, it saves and reuses what works best. And in fact, if you read the papers from the very first study that they did, it was the most surprising finding of the whole study. Not that junk DNA wasn't junk, but that it wasn't junk and it wasn't conserved. Okay? That if it's important, why aren't we keeping it from organism to organism? Right? So they couldn't understand that. And so that's what he's commenting on there. Well, if it's not conserved, it can't be functional. In spite of what they've already found, he's still claiming it can't be functional. And again, they didn't find the continuity among organisms. What did they find? Discontinuity. And that's consistent with the historical science based on God's word that he created animals according to their kind, and we don't have one kind evolving into another. Well-known atheist blogger and evolutionary biologist P.Z. Myers stated this in a blog entitled "Junk DNA is Still Junk." The bottom line, though, is the genome is mostly dead, transcriptionally. The junk is still junk. And it boggles my mind in some ways. How can they keep saying this? I mean, when the observational science is right in front of them, but they still deny it, right? And because it's inconsistent with what they would predict based on evolutionary assumptions. So they are willing to deny it. They're willing to deny the observational science because it isn't consistent and doesn't confirm their version of historical science. But like I said, that's not true of all evolutionists, and some scientists, like the ENCODE researchers and others, are more truthful regarding the discoveries of the functionality of junk DNA and what that means. These researchers who are working on pseudogenes says, but definitely the so-called pseudogenes are really functional, not to be considered anymore just as junk or fossil DNA. It's not. It has a function. Richard Sternberg, in his papers on junk DNA, said, our expectation is that one day we will think of what used to be called junk DNA as a critical component of truly expert cellular control regime. So once again, observational science is consistent with and confirms the historical science based on God's word. Again, clearly design and not disorder. Now, the next part we want to talk about is structural design, and I really had to shorten this section, okay, as much as I would have liked to have shared more. But structural design deals with the physical structure of the DNA, the three-dimensional shape, the packaging of it, and the chemical markers that affect that packaging, and that regulates the expression of the DNA, whether it gets transcribed and translated. But it's based in the three-dimensional structure. We're not talking about the sequence of the DNA here. We're talking about its physical structure and the chemicals that affect that physical structure. So it's a different layer or level of information that is outside the sequence of the DNA. And we're going to focus on just one area of this particular part, and that is epigenetics. And that's because it is hot in the research fields, okay? And I get asked a lot of questions about this, and so I want to discuss it. Now, epi means over, outside of, around, on top of. And that's what this is. It's above the sequence of the DNA. It's not because it's not the sequence of the DNA that's doing this. It's something above it or on it that's affecting it. And so it's this other layer of information. And these are chemical markers and modifications that alter the expression of genes, not by changing the sequence of the DNA, but by changing the availability and the accessibility of that DNA. Okay? So it's another level of regulation affecting the physical structure of the DNA. That's why I said it's not just the sequence where there's information. There's so much more above that, and this is another layer of that. Now you can see here, there's two main areas of epigenetic modification, and you see the little methyl group up there. It's kind of gray with 3 white balls on it. That can affect the expression of the genes. And then you see those little blue things there. The DNA is wound around what are little balls of proteins called histones. And again, that's for packaging reasons. You got to package 6 feet of DNA into this really teeny tiny little nucleus and of all your trillions of cells. So those little balls of proteins have tails on them called histone tails, and those can get chemically modified. There's about 12 different modifications that we know of that can happen, chemical changes to those, like acetylation, phosphorylation, all those big terms that modify that. And when they do that, when those modifications occur, whether it's a methyl group or it's a histone tail modification, it alters the structure of the DNA, which alters the availability and accessibility of it, which alters the expression of it. Okay. Now, so these chemical markers are heritable. And here's where it gets really interesting. Because they didn't used to think it was. You were just thought to inherit the genetic sequence, right, the DNA sequence from your parents. And so people believe that when the DNA from the egg and the sperm united, that all the markers basically fell off or were removed and the baby started fresh. But that's not the case. For all of those markers. It can be short-lived. It can just be passed down from one generation to the next, or it can be longer-lived, being passed down to multiple generations. So you're not just inheriting a genome, you're inheriting an epigenome. Okay? Again, that other layer of information. It's environmentally controlled. And again, whole nother level of complexity. How this happens, we still don't know. How does it know what to methylate, what to acetylate, what to phosphorylate, when to do it? Still don't know. How does it know which ones to do this to? Still don't know. We're still trying to figure this out. We know diet, for example, can affect. So what a mom eats during her pregnancy affects the expression of the genes of her child and can even affect the expression of her grandchildren, okay, the genes of her grandchildren. So you probably heard the phrase, you are what you eat, but you are potentially what your mother and your grandmother ate, okay? So you're affecting by what woman eats during her pregnancy. She's not just affecting the next generation, but she could be affecting who knows how many generations. I want to use an analogy to explain this. I'm going to show you 2 sentences that have the same exact words, but 2 different meanings because the punctuation used is different. Okay, so here we go. A woman without her man is nothing. A woman without her man is nothing. Now, I know, it's kind of funny. Now, what's the difference? And the meaning is totally different there, right? But the words are exactly the same. It's the punctuation, right, that makes it different. And that's what, so again, you have same words and letters, different meanings because of different punctuation. Same sequence and bases of DNA, but different expression because of different epigenetic markers. The epigenetic markers are kind of like the punctuation, okay? So they help decide the meaning. How is it going to be accessible? Is it going to be available? Is it going to be made into protein? For example, identical twins will have the same DNA sequence, but they can have different epigenetic markers, which explains why sometimes one twin will get a disease or cancer, but the other twin will not. Right? And it has to do with that. Now, this next clip I'm going to show you comes from a PBS show. It's actually a very well-done show on the issue of epigenetics, and they show differences in mice as a result of epigenetics.
Well, these mice may hold a clue. Their DNA is as identical as Anna Marie and Clotilde's, despite the differences in their color and size. The human who studies them is Duke University's Randy Girdle. So Randy, I see here you have skinny mice and fat mice. What have you done in this lab?
Well, these animals are actually genetically identical.
The fat ones and the skinny ones?
That's correct.
This gets even more mysterious when you realize that these identical mice both have a particular gene called agouti. But in the yellow mouse, it stays on all the time, causing obesity. So what accounts for the thin mouse? A tiny chemical tag of carbon and hydrogen, called a methyl group, has affixed to the agouti gene, shutting it down. Living creatures possess millions of tags like these. Some, like methyl groups, attach to genes directly, inhibiting their function. Other types grab the proteins, called histones, around which genes coil, and tighten or loosen them to control gene expression. Distinct methylation and histone patterns exist in every cell, constituting a sort of second genome, the epigenome.
Epigenetics literally translates into just meaning above the genome. So if you would think, for example, of the genome as being like a computer, the hardware of a computer, the epigenome would be like the software that tells the computer when to work, how to work, and how much.
Okay, so I thought that was a really great example of that. Those mice can be genetically identical, but look so different because of differences in epigenetics that had to do with actually their diet. And it's known in these mice to be transmitted too for multiple generations as well. In humans, just to give you an example of that, in humans, there is a type of synthetic estrogen that's often prescribed to prevent miscarriages that has now been linked to an increased number of cancers in the daughters and granddaughters of those women that took it, of their reproductive organs. And so they studies point to changes in epigenetic markers related to the development of those organs, which the mothers passed down to their children and then was also passed down to their grandchildren. They found in human twin studies that older twins will have more differences in the expression of their DNA than younger twins, suggesting that environment over time is having more and more of an impact on the epigenetics, that other genome, so to speak, there. So we inherit not just a genome, but an epigenome, not just the sequence of the DNA, but the physical structure of the DNA as it's altered and modified by these chemical markers. So how does evolution account for the epigenome? Because now you not just got to evolve a genome, you got to evolve an epigenome, right? You got to regulate this. And so epigenetics is problematic for evolution because, for another reason, is because evolutionary change is based on random changes. Yet these changes occur in direct response to the environment. That's a big no-no, okay, for evolution. It's supposed to be random, not in relation to the environment. And what controls and causes these changes, like I said, these chemical modifications, we still don't know. I mean, how does it know to put a methyl group there at that time under that condition? What causes it? We don't know. But I can bet they're probably pretty complex mechanisms, right? I mean, think about how many different environments we come into contact with. Think about how many different genes have to be changed in a specific way at a specific time. This has got to be extremely complex. And so these non-random changes imply then that evolution has some sort of mind, right? Because we only want to alter the genes that need to be altered under those conditions. So that's another level of information and mechanism that evolution would have to account for, as if it wasn't hard enough to get the genome. You've got to evolve all these other things as well. Organisms would have to evolve complex mechanisms to make epigenetic changes to adapt to future environmental challenges, right? Because this is something that it hasn't come into contact with, but it's got evolved mechanisms to deal with what's going to happen in the future. Well, where does that forward-thinking design come from? Evolution is mindless. It doesn't prepare for the future. It can't. It doesn't work that way. So it can't see the future, so how could it evolve mechanisms to prepare for the future? I mean, think about it. It can't say, well, gee, in 1,000,000 years, I'm going to face this condition, and I'm going to have to change my DNA to deal with this situation under these conditions. It doesn't know that, right? It's mindless. It can't think that way. So how could it evolve this forward-thinking design? How can it evolve these mechanisms to do that when it's not something that's currently happening, not something it needs to do right now? Because evolution is here and now. It either is something that it can use or it isn't. And if it can use it, select it for. If not, it's selected against, right? That's just how it works. But so it's totally, totally inconsistent with what evolutionary assumptions would predict. But when it comes to creation, it makes total sense, right? Because God's omniscient. He foreknew Adam and Eve would sin, that he would judge that sin, and that the world would be cursed. So God designed organisms with the ability to adapt in a fallen world. Epigenetic mechanisms allow organisms to change easily and quickly in relation to their environment. So if it needs to change again in the future, it can be easily done. You're not changing the sequence of the DNA, you're just changing how it's expressed. And so epigenetic changes, I think, are very valuable because they allow immediate benefits for offspring because it's heritable, right? But they don't ultimately change the sequence of the DNA, at least not that we know of right now. I always got to put that caveat on because, again, we're constantly learning about this and more about this. So again, this is totally consistent with what you would predict starting with God's word and God as the designer and creator of our DNA. It is clearly designed and not disorder. And the last part that we want to talk about here is what I call adaptive design. Now, what I mean by that is the way in which God has designed organisms to adapt. And we discussed one way that this may be accomplished with epigenetics. And in this section, we're going to explore some other possible adaptive designs. Now, another way that creationists have termed this is actually called mediated design. And we have articles on our website that talk about that. That God designed organisms with the ability to use existing parts to adapt to varying challenges. Not changes that would lead them to being a completely different kind of organism, but rather change within a kind. And creation biologist Dr. Todd Wood likes to use the analogy of a Swiss Army knife. So it has every tool a camper might need as he faces the unknown challenge of wilderness living. But he pulls out each tool depending on the situation, uses different tools for different situations. But the tools are pre-existing, right? Something new isn't created, it's just used when it's needed. And so that may be what we're seeing in the DNA, that the information needed to adapt already exists in the DNA or in the organism, but only used when faced with the challenge. And so again, these are not chance changes, not random changes, but purposeful design changes. And there appear to be mechanisms that God has designed in organisms that allow them to respond to the environment and cause specific changes. Now, this is more in the sequence of the DNA that allow an organism to adapt. Now, and many of these adaptive variations may have been expressed after the flood, right, to allow organisms to fill the earth that had many new and different environments. That would be a great advantage to them. One of my favorite examples of this is the iguanas in the Galapagos Islands. And these are pictures I took while I was there of the iguanas. And we have the land iguana and the marine iguana. Now, they have very different diets. The land iguana stays on land, eats cactus pads. The marine iguana, however, is able to go into the water, dive, hold on to rocks, and eat algae, okay, off these rocks in the ocean. Now, which is very unusual it's the only iguana in the world that does this, okay, because they don't normally like to go into the water and get cold because they're cold-blooded organisms. So that's hard for them to do, but that's how they've adapted to that particular area. Now, both have claws, and you can see that in the picture, but the marine, you'll notice, they're longer because they need to be able to hold on to the rocks and eat the algae off of them. They both have salt glands, but the marines are more developed, and they're able to actually sneeze out excess salt that they get by ingesting ocean water. Now, this looks a lot like mediated design, because it's the enhancement or the greater development of existing parts to adapt to environment. Nothing new, just using what's already there in a slightly different way. Now, we have a lot of examples of this in bacteria, and one of my favorites is the long-term lines of E. coli. E. coli is obviously a bacteria in your gut. You also find it in soil. And this is the work of Dr. Richard Linsky at Michigan State University, and he has been continuously growing 12 lines of E. coli in his lab since 1988. So it's a very long-term experiment. He's generated over 50,000 generations of bacteria, and which is equivalent to literally millions of years, okay, if they were out in the wild. And E. coli is a bacteria that is known to use citrate as a carbon source, but only when oxygen is low. Okay, so the bacteria that he started with already did this. Now, around generation 31,000, Linsky found that there were certain mutations in the E. coli that allows them to use citrate when oxygen is normal. So they're still using it, okay, they're just using it under a different condition. Now, this is not the evolution of a new function, but the ability to use a pre-existing function under different conditions, right? It's nothing fundamentally new here. It's just using it under a different condition. Now, how did this happen? And what happened was the CYT gene, which you see in blue there, encodes a protein that transports the citrate, moves it into the cell. Now, it's normally in the top there, in letter A, it's only expressed when oxygen levels are low. But what happened in these bacteria is a portion of that DNA, including that gene, that CYT gene, got duplicated. Okay, a copy was made, and you see it there on the bottom in B. You see two copies. You see two CYTs. And it was placed under the control of a different regulatory area, which allowed it to be expressed when oxygen levels are normal. Okay, when they're not low, when they're normal. Now, there's a lot of citrate in the media that these bacteria are fed, so it was an advantage to the bacteria because now they could grow better than those that didn't have that mutation, because now they could use it under normal oxygen. But again, think about it. Everything was pre-existing. The gene was there, the regulatory region was there. It's now just being used in a slightly different way to preserve or conserve the bacteria in a specific environment. But what do evolutionists claim regarding this finding? The thing I like most is it says you can get these complex traits evolving by a combination of unlikely events. That's just what creationists say can't happen. But it's not the evolution of a complex trait. I've never seen, with this particular set of experiments, people were saying, oh, look, it's a key innovation, you know, and I'm like, seriously? It now uses citrate. It used to use it when it was low oxygen. Now it uses when it's normal oxygen. Ooh, wow. Huge change. No, it's not. It's just using it under slightly different conditions. It's a pre-existing trait under different conditions, not the type of change. That's not what you need for molecules to man evolution. Okay, that's not going to get you to where you need to be as far as evolution goes. And think about it. They've had millions of years in the lab with these bacteria. And guess what? They're still bacteria. Okay. So those are the kinds of changes that we're seeing. And so what we see, again, this is clearly design and not disorder. God has designed mechanisms in these bacteria to allow them to adapt. Nylon digesting bacteria. This is another famous one that evolutionists like to use. So bacteria has been found in wastewater from nylon factories that can digest nylon. Now, nylon obviously is synthetic. It's man-made. It's not a natural substance. So how did it acquire the ability to do this? Well, they found out that there were point mutations, which are just small changes in what is known as the E2 gene, and that produced an enzyme, a protein, that has a different active site. Now remember, the gene is already there, okay? It already possessed that gene. It's nothing new. And you see here in the picture, this kind of showing you kind of an amorphous picture here of the protein. So let's see, on this side, oh, see, people down there don't know what I'm talking about. So the one that has a very U shape, okay, the normal substance can come in, it can be broken down. But then you get the point mutations, and so then you see it has a little bit of a bump now where that U is, and now it can break down nylon as well as the normal stuff. And so, okay, Again, how is that some major change? It's not. The normal E2, interestingly enough, digested a substance very chemically similar to nylon. Right? So in the normal bacteria, with the normal protein, it was breaking down something that was actually quite similar to nylon. Now you have a mutation, an alteration that causes a pre-existing enzyme to digest something that's very similar. Again, that's not amazingly new because everything was pre-existing. The enzyme was already produced by the bacteria and digested a substance very similar to nylon. Slight modifications allowed it to conserve it or preserve it in a specific environment. Now, so there's lots of examples that we could give with bacteria, but we also see examples of what may be mediated design in mammals. Now, obviously, when we move to multicellular complex organisms, this is a lot more difficult to think about because, and to know if this is an aspect of original design or it's mediated design. So I want to put that caveat in there because to change the organism and pass it on to the next generation is going to be very different from a unicellular bacteria, right, passing it on to a daughter cell. You've got lots of cells to change. You've got to change it in the germ cells, the sperm, the egg, and so this becomes more complex, but I still think these are things that are happening, and this is just one possibility of this in what is known as the TRIM5 CYP-A mutation in monkeys. So TRIM5 is a gene that produces a protein that binds to the outer covering of viruses, specific viruses, and prevents their replication, okay, inside cells. Owl monkeys have a unique version of TRIM5. So here's some pictures of owl monkeys, and you can see where they get their name from, right? They have very huge eyes, and their skulls, I saw some pictures of their skulls. They're amazing looking, just huge cavities for where their skulls, for where their eyes go in their skulls. Now, they have a TRIM5 gene that has been fused to another gene called CYP-A. And it forms a protein that is a combination of both TRIM5 and CYP-A. Now, what does CYP-A do to begin with? Well, CYP-A is also a protein that binds to the outer covering of certain viruses, including HIV. Okay? It binds to the HIV virus. Now, we don't know exactly what CYP-A does, we just know it binds. With TRIM5, we know it binds and prevents replication. So what happened in these owl monkeys, at some point, we see what looks like. Again, it could be original design. We have to be very, very careful about that. But what seems to have happened is that the CYP-A gene, and again, we're not going to go through exactly how this occurred. It's called retroposition, but got put in really close to the TRIM5 gene, producing an RNA that was a combination of TRIM5 and CYP-A. Then that got translated and made into a protein that was the combination of those two proteins. So it's called the TRIM5 CYP-A protein. Now, that protein has antiviral activity of TRIM5 coupled to the HIV recognition of CYP-A. So this prevents infection in owl monkeys by HIV. They can't get HIV because of this. Because of that combination, because of that fusion protein, it prevents them from doing that. So again, if this truly is mediated design, it's the fusion of two pre-existing genes producing a fused protein that has the same function, but slightly different specificity. Right? That's the only difference. Just like with the citrate, it's just doing it under different conditions. Now it's just doing it against a different virus. Again, everything was pre-existing. It didn't change the function of TRIM5 or CYP-A. It just allowed them to recognize a different group of viruses and exert antiviral activity against those viruses. So we have slight modifications to preserve or conserve the monkeys in an environment where HIV is prevalent. Now, this is, again, I think, a good candidate for mediated design. It doesn't appear to be random chance fusion, rather a design response to an environment to allow it to adapt. The molecular mechanism, how this exactly occurs, how it knows to do this, very much like in the bacteria, we don't know. But what do evolutionists claim regarding this finding? Here, a single mutation has resulted in a new protein with a new and potentially vital function. New protein, new function, new information. No, it's not. It's two proteins that it already had. It just fused them together, and now it allows it to recognize a different group of viruses and do the same thing that it did before with a different group of viruses. Again, that's nothing really new there. That's not the type of change you need for molecules demand evolution, where you've got to gain structures and functions and completely new things to go from one kind of organism to another. If these are only the kinds of changes we see, you're never getting anywhere, right? You're never going to go where evolution needs to go. So this is clearly design and not disorder. So again, the changes that we have discussed do not seem to be the result of random genetic chance genetic changes. I mean, think about it. How can an organism wait around, right, for the trial and error mechanism of evolution? How is it going to wait for these things to occur by chance? But rather purposeful design changes that allow organisms to adapt and survive. What the mechanisms are that accomplish this and how this gets passed to the next generation are still unknown. But look at all the levels of regulation that we're only now beginning to discover. I mean, think about it. What I shared with you earlier, no one thought we had dual coding genes. No one thought we had duons, dual coding codons. We're just beginning to discover all these levels. And I think a lot of it is because for many years, people never bothered to look there, because they just thought it was much more simplistic than that. And we've avoided it because of that. So again, adaptive design is inconsistent with predictions based on evolutionary assumptions, but consistent with what we would predict starting with God's word and God as the designer of and creator of our DNA. As Ken shared, there's no question that we can know God through what he has created. Romans 1:20 says we can know him through his creation. People are without excuse. All these geneticists that look at this, that are evolutionists, are without excuse. So if design is so clear, again, then why do many scientists still not believe God is the designer and creator of our DNA? Because people suppress the truth in unrighteousness. We know from Scripture that it's not a matter of not knowing. They know it in their hearts, but they have suppressed the truth to continue in the rebellion against God. They continue to deny the obvious. And that's why it's so important in our apologetic that we never forget that this is not about the evidence, but rather it's about our starting point, our presupposition, our foundation when we look at that evidence. Is it man who wasn't there during the supposed millions and billions of years of Earth history? Or is it God who was the eyewitness to creation and inspired men to write that down? And we know, like Ken said, we can know God through what he has created, but may we always remember that faith comes by hearing, and hearing by the word of God. And I want to help you know how you can become equipped to think about some of these things a little more and be able to give the answers. I chose the topics I did because they're the things I hear about the most out there. I have several DVDs that relate to these kinds of topics, like genetics, evolution, and creation, most asked question. Wonder of the Cell talks a little bit more about cellular complexity. And then the Code of Life, DNA, information, and mutations, looking at mutation and natural selection.